So this was this other talk that I gave is our pilot study looking at the phosphorylation state and post-translational modification state of different proteins found within the serum of migraine patients as compared to controls. So just in brief, to have a little background. So there's been a lot of studies looking at genetic susceptibility to migraine and looking at things like genomics or genome-wide association studies to identify specific DNA modifications that may make a person more susceptible. There's also been some work done in terms of the transcriptomics, so looking at the RNA level. So we've got DNA level, RNA level, and correlating those changes in the migraine patient from plasma, CSF, all sorts of different tissues to say, okay, what is the transcript of these patients? Does that again help us determine disease state or severity? Those studies have been correlated with imaging studies, but and then there's been some other movements towards metabolomics. So again, lipid studies, as well as what we call cytomics, which is looking at individual cell type contributions by isolating things like neurons or astrocytes or microglia. My lab was really focused in on the proteomics, and so looking at the protein level, because protein is really what's being manifest to cause changes in functional outcomes, right? And the way I illustrated, and I like to illustrate it, is that if you look at the genome and the genomic studies, that's like having a map in front of you, right? It tells you where all the things are in time and space, right? If we look at the transcriptomics, that's like you sitting down, looking at the map and saying, I would like to plan a trip and highlighting the roads that you want to drive on, right? Proteomics is really getting in the car and starting that drive, right? So you're actually doing the thing. So to take it one step further, if we keep on this driving and map analogy, right? Just because you're driving a car doesn't mean that you're going to get from point A to point B in the 5 hours that you've allotted. There are things that can come up. There are traffic lights. There are construction zones. There may be weather advisories, right? You might run out of gas. There's a lot of different things that happen. And so that's really where the post-translational modifications and the phosphoproteome comes into play. And so just like a traffic light will kind of tell you when you can and can't go, the phosphorylation on a protein gives it a level of activity or decreases its level of activity, as well as these other post-translational modifications, including things like oxidation and acetylation. So the first take-home message from this particular talk was that we could look at the total proteome, so just what proteins are there, and we could identify chronic migraine patients, or just migraine patients in general, from the healthy controls. When we dove deeper in and we looked at the post-translational modification, we found that under the normal state, most of the proteins that were identified in the serum of controls had a phosphorylation group. In comparison, the migraine patients, most of those proteins were actually oxidized, suggesting that there was cellular stress. Right? When you looked at then comparing the two oxidate, because all three of these modifications are present in both. So when you actually look at the signaling pathways that are implicated in the oxidized state, it was changing protein phosphorylation. So that's why we kept going with the phosphorylation component. What was really interesting in the what we call the phosphoproteome or proteomic studies is that we were able to identify 3 proteins that had very, very clear phosphorylation state differences between the migraine and the control patients. The first 2 were really implicated in, one was fibrinogen alpha, and the other is called sushi domain containing protein 5. And the phosphorylation sites were present in all of the control patients and completely absent in all of the migraine serum. Why is that important, right? So those particular phosphorylation sites, because proteins are big, they can have many modifications going on at different points. But the particular sites that were missing in the migraine patients were related to extracellular matrix organization and cell adhesion, suggesting that they might have deficiencies in being able to maintain this the space in between cells and how the cells are connecting to each other. On the flip side, we had another protein, the third protein, that was largely absent from the controls and highly significantly upregulated in these migraine serums. And so this was a protein called band3. It's an anion transporter that maintains balance between anions and bicarb. And but the phosphorylation site that was present in the migraine patients and absent in the controls is again implicated in cell adhesions. So again, going back and suggesting that maybe there's a problem with not just these signaling cascades, but how the cells are actually maintaining their organization relative to each other, right? And when you look at things like a proteome or a genome or the transcriptome, the ideal is that we're trying to find a biomarker. So something that's accessible, reliable, and relevant to your disease state so that you can track, disease progression, disease severity, and treatment response over time, right? And so that's really where we see this kind of going is now can we take some of this information, validate it with some more traditional techniques. So this was all done by quantitative tandem mass spec proteomic analyses. But can we actually either add patients? Can we go back to these same patients after they've had different interventions and see if there's restoration and if that corresponds to their headache pain, right? To really move forward with that. The other part that we see this as is we were able to do some secondary analyses and we're able to identify within using these post-translational modifications, patients who had episodic migraine versus those who had chronic migraine. We were also able to take the same data, again, the same data, it's just these multiple layers of analyses, and identify those patients who had aura versus without aura. And so it really allowed us to identify, even though the proteins were the same, when you looked at when and where they were phosphorylated, the pathways that were implicated were different. And so we were able to use that to validate some of the genomic and transcriptomic and imaging data showing neurovascular challenges, as well as these extracellular matrix challenges, as well as confirm some early studies from the 1990s, suggesting that the complement cascade and immune responses were really engaged during migraine. We were able to identify not just this cascade, but where in the cascade we were seeing these changes. And so, these again are pilot studies, but it seems to be that we can actually utilize these post-translational modifications to make some inferences about disease state, about disease severity, about disease progression, because we could identify the episodic from the chronic. And then be able to use that information to maybe develop more selective interventions. So things that maybe can prevent the transition from episodic to chronic, or, that maybe have more effect in an aura patient versus a without aura patient.